uyarımız da geldi. Peki. İyi akşamlar herkese. Bu akşam Pınar Önal'ı hem tanıyacağız hem kendisinin araştırma alanlarını dinlemiş olacağız. Ben sözü direkt Pınar'a bırakıyorum. Hoş geldin Pınar. Söz sende.
Teşekkürler Efe. Hoş bulduk. Merhaba herkese. Ben Pınar. Bilkent Moleküler Biyoloji ve Genetik'te Eylül 2022'den beri asistan profesör olarak görev yapıyorum. Konuşmaya başlamadan önce biraz kendimi tanıtayım. Bu arada şimdi Türkçe konuşuyorum ama konuşmayı İngilizce vereceğim. Bunun için de önden bir özür diliyorum. Benim için daha kolay olacağı için bunu seçtim. Çünkü zaten bu çalışma hep yurt dışında oldu. Türkçe konuşmakta biraz zorluk çekiyorum yeni döndüğüm için falan derken. Öyle olmasını seçtim ama tabii ki Türkçe sorular soracaksınız siz YouTube'dan artık. Türkçe cevaplamaya çalışacağım sunumun sonunda. Dediğim gibi daha yeni başladım Bilkent'te. Öncesinde postdoc için Amerika'daydım. Uzunca bir süre postdoc yaptım. Önce NYU'da Drozofila çalıştım. Daha sonra Northwestern'a geçtim ve bu çalışmama devam ettim. Geriden başlıyorum. Doktoramı da daha farklı bir organizma üzerinde yapmıştım. Schmittea mediterranea, flatworm ya da yassı solucan de denilen bir canlı, rejeneratif kapasitesi çok yüksek olan bir canlı. Orada başlayan evrim serüvenim daha sonra Drosophila development'da Evo halinde alarak devam etti. Şimdi hala işte evrimci, development ya da gelişimci olarak çalışmalarımı sürdürüyorum. Daha fazla uzatmayacağım ben. Hemen sunuma başlıyorum o zaman. Konuşmamın başlığı, buradan İngilizceye çeviriyorum ben hemen. Çünkü bundan sonrası Türkçe biraz zor olacak. So, today's topic is about, or today's the talk's title is Evolution of Novelties by Changes in Transcription Factor Sequences. And that's a work that has been done, or that I've been working for the last couple of years, starting during my postdoc in US and now here in my own lab. The work that I'm going to present to you tonight will be a work that was done by several people, from undergrad students to professors. So it's a collaborative work, and most of the work that work that I'm going to talk about will be published, and they are available on open access journals. So if you can't, if you don't understand, and if you don't have a question immediately, then you can go online and find those publications. So as I said, I'm an evolutionary biologist, developmental biologist. More likely, when you see the talk, maybe you will say, what does this have to do with evolution? And then I want to tell you this. I want to give you a quote from Conrad Hall-Weddington, one of my scientific heroes, where he very beautifully puts my feelings into words here. It's a long quote. I will summarize it for you. Basically, what he's saying here is that since evolution is concerned with the changes in animals, it is impossible to discuss or to understand evolution without understanding the system. And that's why I studied development, to understand these developmental pathways and to see how a developmental pathway can be modified. To me, evolution is more than numbers, it's the development itself. So that's why I study development, and that's the model that I study with fruit fly, Drosophila melanogaster. And I work with other models. And when I say this is the best model, you have to trust me. I know I might be offending people here, but I think fruit fly is one of the best models. Well, I'm downgrading it from the best to one of the best models for several reasons. First of all, it has a short generation time. And this is very important if you study development, because you basically can watch an organism grow under the microscope. That's not really possible for most of the complex organisms. And they have high fecundity, which means a female in the peak of its fertility can lay up to 50 eggs. Again, you can lay eggs, you can collect eggs several times a day. So they don't have a seasonal laying schedule or anything. So they can lay eggs from early morning till evening. So you can collect a lot of embryos, your sample or my sample, to work with the whole day around, like hundreds of them. Another thing is that they have a very highly annotated genome. So you know where your gene of interest is, you know what other gene is, how far it is from that. And based on this knowledge, you can act on by the availability of a tremendous amount of genetic tools. Anything you think of, and that's possible in Drosophila. It also shares 60% of its genes with human, which means that it has also some relevance to human biology. And it is a simple organism, but it has a very complex molecular function. So it is important, and it is very informative understanding basics of human biology. Last but not least, it also is cheap to maintain, which is important in a country like this, I guess. This is a recent picture from our fly lab. You can see here in these two. Let me turn on my laser pointer. These two shells contain 200 transgenic flies with duplicates. And these are our vials, which have fly food based on sugar, yeast, and flour. So they're really very modest. So they don't ask for much for what they do, what they provide us with the whole information and data about biology, about life. And it's not only us or me who likes Drosophila. It is there's a big fly community out there studying on this. So there are a lot of flies. If you can ask. You can ask for help. You can ask for flies. So it's a very cool system. And also Nobel Committee acknowledged several times or knows that Drosophila is a great organism. Nobel Prize has been given to Drosophila scientists at least 6 times. And here you are seeing 3 Drosophila scientists, developmental biologists, who got a Nobel Prize in physiology and medicine in 1995 for their work on the genetic control of embryo development. So that's I'm showing you this because I also work on embryo development, and these are the people that I can relate to more than the other Drosophila researchers who can be working from anything from circadian rhythm to nervous system and so on. And I work with changes in transcriptional regulation and how they affect phenotypic differences. And transcriptional regulation is mediated by transcription factors. So the main actor of the transcriptional regulation is transcription factors. I think nobody in the audience needs this introduction, but we'll briefly mention what they do, how they recognize. So transcription factors are composed of at least one DNA binding domain and several activator domains. And together with the activity of these domains, these proteins recognize specific sequences that are called motif and that are found across the whole genome that are 6 to 8 base pair long. These transcription factors find the sequences and bind these sequences in the right time and space to recruit cofactor and transcriptional machinery to regulate the gene activity of downstream genes. And any change in the cis-regulatory or trans-regulatory sequences, I mean in the DNA sequence or of the motif or the regulatory region or transcription factor, either in the DNA binding or activator domain, can lead to changes in DNA binding preferences or cofactor binding preferences, which might most of the time lead to morphological abnormalities, but these can be also used as a way to create novelties, variations by evolution or by developing organisms. So I am mostly interested in this transcription factor sequence changes. And the transcription factor that I'm going to talk about, and I have been working on, is bicoid. I guess, again, it's a very famous protein. It's one of the first morphogen identified. I already gave you the next thing that I'm going to say. So it's a maternal factor. That means the mother transcribes it and loads it to oocyte anterior, the transcript, and upon fertilization, the transcript is translated, and this this protein forms an anterior to posterior gradient, which is important in the temporal and spatial regulation of the target genes along the AP axis. And these are all very highly conserved genes that are found in vertebrates, in human too, homologs of which are found in human. Bicoid is a transcription factor, so it binds DNA, and the DNA binding domain of bicoid is a homeodomain. For people who might not know homeodomain, it is a very highly conserved, highly famous DNA binding domain. It is, I think, found in like 200, maybe more proteins in human and hundreds of proteins in Drosophila. It's highly conserved, as I said, from yeast to human. Bicoid is so essential for the embryo. Here you see on the left-hand side a wild type. Why is it now? Okay, wild type embryo from the start of cellularization till the beginning of gastrulation. There's a 2-hour time window, and from here to first instar larvae it takes another 15 to 20 hours. So this is the wild type embryo, and you can see the anterior posterior difference. And this is a first instar larvae that is formed in less than 24 hours, and it has 8 abdominal, 3 thoracic, and several head segments, anterior to the left, posterior to the right, and that will be the orientation that I'm going to use throughout the whole presentation. And here is the bicoid mutant embryo, and I will show you the video of it too, and you will recognize right away what is wrong with this embryo. As you can see, the polarity is lost, and here in the larvae it turns into a complete mass. There's no anterior structure formed, and posterior structure, which is labeled here as FK, which is Pilz-Körper, formed in the anterior. So bicoid is required for, and also sufficient, that is what I'm not showing here, for the embryo development. despite being such a critical protein and also regulating a very conserved set of downstream target genes, bicoid itself is not a very old protein. So it's called a novelty. Well, this novelty has been around for around 100 million years, but it's in the insect phyla, it's really found in a subset of flies, including Drosophila and some closely related species. What is believed to happen is that in an ancestral HOX3-like gene, which is found in HOX locus, which I referred here as ancestral Zambicoid, a gene duplication happened. And that led to, that's why I call it ancestral Zambicoid, that led to the formation of Zen and bicoid proteins. So these are paralogs found in Drosophila and some closely related species. So this gene duplication event, they are very critical for speciation, for the formation of novelties. And in this case, it also is very important, and it led to formation of these paralogous genes, which evolved into a complete, like, exclusive expression domains. So, and related to that, they also have different functions. Zen is expressed dorsally and expressed later on during embryo development, so zygotically expressed in the dorsal part of the embryo, and is involved in extra-embryonic tissue development, and it is believed to be a more ancestral role. Whereas bicoid gained its novel anterior functions. As I said, it is maternal and distributed along the anterior-posterior axis in a gradient. So when this project first started, the question was very simply if they do share, whether or not they share any functions. So because we know that they are involved in different functions. One is expressed later and one is expressed earlier by the mother, and they are expressed in different locations. What happens if we express then maternally and see if it can rescue bicoid? And to do that, we basically constructed this vector that has bicoid regulatory sequences, bicoid 3' UTR and bicoid promoter, to ensure maternal expression and anterior localization of any gene that is expressed. We also wanted to have a control that this construct works, basically by expressing bicoid in the same construct. And we expressed these these transgenes in bicoid mutant embryos, and then to look for if they replace any bicoid functions. So the control worked. So that was like, you're good to go. You can trust this construct. The regulatory sequence are working, and we can rescue bicoid mutant with the bicoid expressed from this construct. And we tried it with the Zen. I call it maternal Zen because it was expressed by the mother. And unfortunately, this was a complete bicoid null phenotype. As you can see, Filskerper in the anterior, this is a lethal phenotype, so it doesn't really rescue. So Zen does not share any bicoid's anterior faction. So how did bicoid gain novel anterior functions then? When you look at the sequences when you compare the sequences of these two proteins, you see that they really diverge a lot. So there's no similarity outside of the homeodomain. And even in the homeodomain, they share only 43% similarity. And homeodomain is a 60 amino acid long sequence. So this is really a very striking divergence in the sequence. And one has like they have very different binding motif. I know they don't look very different, but TAA-TCC and TAA-TTA. clearly are very different. And they do have very different, they correspond to different homeodomain families as well, these motifs. So we wondered if homeodomain can be responsible for this evolutionary divergence. And to answer that, what we did was basically replace Zen homeodomain for bicoid homeodomain. And we also did the reciprocal swap. So replace a bicoid homeodomain with Zen homeodomain. Here you can see these constructs, again, in the same maternal expression constructs, and we are still expressing these in bicoid mutant embryos to observe any change in the anterior function. If we don't supply it with any endogenous bicoid, it can be provided with this construct. And here you can see that Zen homeodomain is complete. It's a completely non-functional homeodomain when it comes to anterior patterning. This is again bicoid null phenotype. These embryos are lethal, and this, however, is a very significant increase. And I have to again remind you that the whole protein is 300 or 400. I can't remember exactly now, but around that range. And the homeodomain it's just a 60 amino acid long sequence, and we replaced it with that. And we saw, and you have to compare this phenotype with this. So this basically told us this was basically able to construct all the abdominal segments, all the thoracic segment, the 3 thoracic segments, and head segments were partially rescued too. So this told us that homeodomain is necessary. That was clear. We knew it from previous also rotation analysis for bicoid. Yes, for bicoid. And we also showed that it is almost sufficient. And we were kind of satisfied with this result. And we asked the next question then from bicoid, how did bicoid evolve? Now the question became, how did bicoid homeodomain evolve? And this simplified the question and made it more accessible through ancestral reconstruction analysis. So what this is basically predicting, by using different statistical models, predicting the ancestral sequences that the bicoid and Zen homeodomains evolved from. And we collaborated with Joe Thornton, which is an expert in his field in University of Chicago, to predict the sequences. Here you see a phylogenetic tree. There are a lot of, so they gathered 33 species, the homeodomain sequences of Zen and bicoid protein, as well as HOX3 proteins as outgroup to basically create this phylogenetic tree and to predict the ancestral Zen bicoid and ancestral bicoid. So ancestral Zen bicoid is the ancestral sequence just before the duplication event that gave rise to ancestral bicoid and ancestral Zen. And ancestral bicoid is the ancestor to all the existing bicoid homologs, basically. And what you can see here by a quick look at this tree is that all the Zen homologs are clustering very close to the ancestral Zen bicoid, showing a very low evolutionary divergence or change here, whereas ancestral bicoid is having this long branch, indicating again a lot of evolutionary changes leading to ancestral bicoid and bicoid homologs as well. So then you might ask what the difference are, and the rest will basically be about the differences between these homeodomains. So these homeodomains are very different. It was also very obvious in the phylogenetic tree. In the difference, if I can summarize you here is there are 31 amino acid difference. The ones that are labeled here are all different residues between ancestral Zambicoid and ancestral Bicoid. So let me just walk through this slide a bit. I call these subdomains N-terminal on helix 1, helix 2, and helix 3. I'll show you the structure. I had to have it here, but the slide is too crowded, so I put it in the back. So homeodomain is a helix 2 and helix structure with an N-terminal arm. And these are, I call them subdomains. And as you can see, the evolutionary or the changes, those evolutionary changes happen all across the subdomains almost equally. Well, maybe a little bit less in the recognition helix. It's the one that binds to the major groove of DNA. And the ones that are highlighted here are diagnostic residues. What diagnostic residues are basically the ones that are found in the Bicoid, ancestral Bicoid, are only found in the Bicoid lineage and never found in the Zen lineage, vice versa. That means that they are more predictive in the functional evolution of Bicoid than most of the rest of the residues, the changes. So we tested the function of these proteins, obviously. So the first one I'm showing you is a result of protein binding microarray. Protein binding microarrays are a way to detect transcription factor binding motifs. So basically they are composed of, it's a microarray and it has all sequence variants of 10 mers, DNA oligomers. By basically incubating those microarrays with your protein of interest and detecting that interaction by antibody bound to your bound protein, you can basically have an estimate about the binding frequency or binding affinity of the protein to the specific sequence in that microarray. And then from that fluorescent intensity, you can calculate an E value, which is also correlated with the affinity of that protein towards that sequence. Here on the y-axis, you see Drosophila melanogaster Bicoid homeodomain plotted against ancestral Bicoid homeodomain. There's a very high correlation suggesting or indicating that they have very similar binding preferences. They both have TATC Bicoid motif as their highest binding motif. So this was good, but we tested also in vivo. And in vivo, it's the same way that we did maternal construct, and we remove Bicoid homeodomain and put our ancestral homeodomains, predicted ancestral homeodomains, and express them in Bicoid mutant embryos and test the functionality. And these embryos do not only form a fully functional or fully formed larvae, but they also can develop into adulthood, and we can keep these animals. So this is basically I don't know if you can call it a new species, but an ancestral, partially ancestral fly, which is completely functional or viable. On the other hand, ancestral Zambicoid showed a very big divergence from the binding pattern or binding profile of ancestral Zambicoid. They have a very low R value, which means they prefer different binding sites. And again, ancestral Zambicoid recognizes or prefers Zen motif, whereas Bicoid, again, ancestral Bicoid binds Bicoid-like motif, which was also not very surprising. And that is also confirmed by the embryo data or the larva data, morphological data. So this homeodomain fails to rescue any Bicoid function. So now we have these two relevant ancestral nodes or proteins. Now the question is how ancestral Bicoid evolved from ancestral Zambicoid. And so the first candidate that we tested was the 50th residue, the change in the 50th residue. Anybody who works in homeodomain knows about this 50th residue. That's one of the most critical. There are like, there are, I guess, 4 or 5 residues that are very critical based on in vitro data that are very critical in the determination of specificity, binding specificity in homeodomains. And 50th residue is one of them. And this is one of the also diagnostic. It is blue. It is a diagnostic residue. So this K is never found in any Zen homeodomain. And we basically asked if changing that residue would change the binding affinity and the function. So first I will show you ancestral bicoid. So K is lysine, Q is a glutamine. These are very different residues chemically as well. So ancestral bicoid converted, whose K is converted to Q, has a very similar binding pattern as ancestral Zen bicoid, like the CNA binding domain, Zen-like motif, and also has lost its function. This was fully functional. This is the protein, and this is single amino acid change, and it becomes completely non-functional. And here is our Q50K change. So this time, non-functional ancestrals and bicoids, glutamine is converted to lysine. And again, we saw a very high correlation between the binding patterns, binding profiles of these ancestral bicoid, now ancestral Zen bicoid. Q50K has a very similar binding pattern as ancestral bicoid. And what we expect was to have also a very significant functionality in vivo. However, what we saw was quite a disappointing image. So this is ancestral Zen bicoid, and you can see clearly that posterior formation is suppressed, but it is nothing similar or nothing as striking as this binding profile change. So I'll come back to that. For now, just remember that that was enough. So that's not the end of the story for us. We wanted to look for the in vivo requirements. I'll come back why they are different later. But let's go on to look for the in vivo requirements. And going forward, I also will show you the transcriptional or expression pattern analysis that we did to be able to be more quantitative, because as it made progress in the function, we also saw very similar expression patterns, which I'm not going to show all, but we had more quantitative analysis by looking at the expression patterns. So here are the 3 previous mutants that I showed you. Ancestral bicoid is completely functional. The expression pattern is like wild type. It is great. This is our fully functional mutant or ancestral protein, and this is the non-functional one. And you can see that none of the bicoid dependent expression is active here. And the K50 mutant, so the single mutant that I showed you in the previous slide, which suppressed the posteriorization, have only these 2 genes activated. And I'll talk about hunchback. I'm talking about hunchback. So hunchback is a very conserved and also a very important anterior patterning factor. Is one of the first and also highest affinity targets of bicoid. And also Eve is another homeodomain protein that is a target of bicoid and involved in the basically patterning of the anterior posterior body segmentation of the body plan, and also separation of thoracic segment from the abdominal segment. so the next candidate that we tested is the R54. So again, it is the 54th residue. It's another important residue that we have known previously from the previous mutation analysis on Bicoid-2. It is not absolutely necessary for the survival, but it is kind of it's a unique residue that is found together with K or lysine. So K and R, lysine and arginine, are not found in any homeodomains in the whole or in the known homeodomain sequences, but only in Bicoid lineage. So we thought maybe changing these two together can confer extra functions. And indeed, we have seen an improvement in the segmentation. Now we have 3 thoracic segments formed, and we also saw some anterior segments being formed. And we had increased expression of hunchback, and OTD also is expressed. OTD again is found in mammalian, in human. It's a gene that's involved in the brain formation, brain development. And again, E2 expression is also kind of increasing with this another change in the amino acid sequence. However, this was also not enough. I did many more, but I want to show you that I also did all diagnostics. So all of the 11 diagnostic residues I changed. And again, we saw all of these genes expressed. This time, all of the tested genes are expressed, but expression itself wasn't enough because it really did not reach to the pattern, wasn't right. This posterior boundaries of these genes did not reach to the posterior boundaries of the wild type sequences, and relatedly, the embryos or larvae could not fully form. There are still some missing head segments here, and this was still a lethal phenotype. Again, as I said, I tried other combinations, but I'm not going to bore you with those details. I'll just tell you that after that, we decided to change our approach to this. We said, okay, let's just swap these subdomains and see which subdomains are leading to a functional protein. Coming back to the subdomain, so this is this is the predicted structure of Bicoid bound to its motif. As I said, it is a helix-turn helix structure. It has 3 helices. Recognition helix is the third helix that directly interacts with the major groove of DNA. And the N-terminal arm is another critical subdomain of the. All are critical, but N-terminal arm is the other subdomain that binds to DNA, or that interacts with DNA directly. And this time, it basically interacts via minor DNA. So, here are where I left you. I tried others, but I have. In the recognition helix, we had K50, which did not really change a lot. So I then replaced the whole recognition helix. And this is the major groove interacting subdomain. And I checked the function again. And here you can see quite a significant increase compared to just K50, and more than K50 are 54 in some other that I made, but this was still not rescuing completely to adulthood. These embryos are lethal, and then I tried to combine two DNA interacting interfaces, or 2 DNA interacting subdomains, recognition helix and then N-terminal arm. And this led to formation of several wild type looking larvae. So this is first instar larvae, and we also saw like very, very few adults coming out of this embryos, less than 10%, and it's most of the time around 5%, not more, not close to 10%, but 5 to 10% of the embryo can make it to adulthood. And I have to remind you here that these are all coming from an isogenic line, which I will refer again and come back later. And the next, I tried also other subdomains, and Helix 1 gave, again, a fully rescuing larval phenotype. Around maybe 20-30% of the larvae could form fully developed structures, segments, and again, less than 10% could survive to adulthood. And I have to say, these are not doing the exact same things. So or that's my assumption, because when you look at the larval morphology, you would see different phenotype, different distribution of phenotype. So that indicates that with these different subdomains, homeodomain is able to do other things, and the combination of them increases the function and increases the survival rate, increases the larval morphology, and leads to around 40% of survival rate, which was still less than wild type, and also based on molecular studies, it wasn't really as good as wild type, which we did not expect, but it was still a survival with maybe slightly different mechanisms. So with this, we were basically able to show progressive and alternative phase of functionalization of bicoid homeodomain. So this basically is maybe those 10% surviving mutants are kind of a bottleneck that a new developmental system or gene regulatory network can be built up and on. This is not all optimized, and none of the changes, none of the mutations, or most of the mutations are not welcome in animals. So this can be used in the future as a way to synthetically change and figure out what are most important, what are the minimum set of genes required for embryo patterning. And again, as I said, synthetically design a regulatory network in the embryo. And these are the things that we are doing, or we are planning to do in my lab. In addition to that, though, we got curious about this and why there was a difference. If you remember, I showed you this striking change in the protein binding microarray with a very unstriking in vivo function. So why is that? Maybe it is not. It's not like a brainer to say eukaryotic transcription factor binding and activating are not 100% correlated. So binding does not mean activation in eukaryotes. It can bind but not activate. It can bind but not enough to rescue, or it might not bind because a binding array and a nuclear environment are completely different setup. And also it was a homeodomain that I used, homeodomain alone. In the in vivo system, it is a whole protein. So which one is the answer to that, we wondered. And we did some experiments to address that question. Basically, CheapSeq to detect the binding, in vivo binding for these different proteins. So these are for people who don't know what CheapSeq is. I suppose most of you know, but basically it is chromatin immunocrystallisation combined with high throughput sequencing, what it is you basically target a transcription factor of interest after fixing it to the DNA, to the chromatin, and then pull down that chromatin bound to that protein, get rid of all the proteins and also excess DNA, and then map this sequence to the genome in order to identify what this protein bound. So you get some peaks and stuff when you map it back to the genome. And that's what we did. And these are the flights that I used to do this analysis. Ancestral bicoid, Zen bicoid Q50K, the one that I saw a lot of change in vitro but not in vivo, and the bicoid K50Q, the one which lost all its function by a single mutation. And bicoid mutant embryo, which doesn't have any functional bicoid, so it serves as a background. And I'll do the analysis based on a log-fold change compared to this bicoid mutant embryo. So do mutants bind bicoid pigs then? Because that was why we did this analysis experiment. So here are two very high affinity bicoid target genes. Hunchback, I showed you before, it binds hunchback proximal enhancer. Here you can see that is where bicoid bind. These are log twofold change. Again, look, ancestral bicoid versus bicoid mutant. It is highly enriched in this promoter enhancer sequence, and it drives this anterior expression. And our Q50K mutant binds very, very weakly, but still drives expression, the one that I showed you previously. K50Q loses this binding completely. And as you can see, there's no expression. And the other enhancer that I checked is Knopf's posterior enhancer. It's again another very critical bicoid target gene. It has many human homologs too. It is expressed in this pattern in the wild type and bound by bicoid. It's a very, again, high affinity and one of the first targets of bicoid. Again, ancestral bicoid binds, Q50K does not. Oh, well, sorry, Q50K binds, but very, again, weakly, stronger than hunchback, but still much weaker than ancestral bicoid. And we see some expression that is most likely the result of this binding. K50Q does not show any activity. This is all background. And in this binding, although we see some sort of binding, the pattern is very different. And also it's non-functional even if there is any binding. And it's very, very, very highly diminished compared to ancestral bicoid. What about novel binding? Well, Q50K was a very weak binding. So it didn't have any novel bindings, but yes, Q50K. K50Q, on the other hand, this is the one that converted binding affinity from a bicoid motif to a Zen motif. And if you remember, so this is a dorsal gene, and if you remember, Zen is a dorsally expressed gene. And we saw this paneer, a gene called paneer, which is expressed dorsally, and all of a sudden K50Q mutant of bicoid starts to bind to the sequence, which was kind of interesting. And we saw a similar pattern with this time a different dorsoventral gene called pyromus. Again, these are not called as pigs in bicoid wild type or bicoid bicoid wild type, but they are novel pigs that are generated by this mutation, or the new specificity led to binding in this new enhancer sequences, which was pretty exciting and interesting, which we want to see if these really are driver of a novel or which if they are real by doing reporter assays and so on in the future. And we also checked the genome-wide profile differences. As I said, Q50K. So on the left, you see a Q50K versus ancestral bicoid, and as you can see here, there's really very few enriched peaks in Q50K, and most of the time, this is the all peaks that are found in all of these 3 mutants. So most of the time, they are not found in Q50K. And when they are found, they're very weakly bound, and they only bind a subset of bicoid targets. So maybe to actually, at the end, they bind bicoid targets, but mostly high affinity targets, and very, very weakly. So the question if whether or not binding is mostly no binding, but when they bind to that TAA-TCC containing high affinity binding sites. As you can see, these are the highest affinity binding targets. They are bound, but they are significantly downregulated in the Q50K mutant. However, K50Q was different, although it has a lot of, it was completely non-functional, and it gained a lot of novel binding affinity. So we saw all these dorsoventral genes, some posterior genes being bound by this novel, by this mutant. And at the end, but it failed to bind to the most significant bicoid targets. A few of them were not bound by this protein, or the highest affinity targets were not bound by this mutant. So at the end, this protein shared more targets with bicoid, although non-functional, shared more target with bicoid, but failed to recognize and bind the highest affinity of those, which kind of was a surprising finding for us. We really thought of not having any binding to any of the bicoid targets, but I guess there's still similarities, and the differences are based on the very highly specific and critical targets. So by the end of this, we were completely, I think, convinced that in vitro binding specificity does not really mean in vivo functionality. And also, since, in vivo, a developing system can find its way around. It's not like a microarray trying to find the binding target. So the next question that I'm trying to answer right now is how specific in vivo transcriptional activity is determined. And in vivo transcriptional activity is determined by modulatory cis-regulation, cofactor interactions, flanking and interacting far-away sequences, and instantaneous epigenetic landscape. That was very clear from our results, and not only from our results, but that is what is known. It's very well known that in vivo is a very nuclear environment that affects the transcriptional activity, transcriptional regulation. And there is a lot of work done in modulatory cis-regulation. Cis-regulatory evolution is basically dominating on the field of evo-devo. So I tried to focus on these other 3 that are mostly ignored, recently ignored, at least. There were a lot of work done on activator domains, and that is also what I'm going to focus in my lab. Basically, this is a work not only done by the DNA binding domain, but involves activating domain, which are mainly intrinsically disordered regions. So I'm moving a little bit. I'm not going to spend so much time, and it's the end of my talk, but IDRs, because I want to introduce this topic, because that's what I'm going to work in my lab. These are basically what we used to call activator domains. They are intrinsically disordered. They're not very conserved. They do not fold into a stable structure under native conditions. So it's hard to predict their structures. And if you look at an AlphaFold or any protein, if you put your protein sequence in any protein prediction algorithm, they will give only your DNA binding domain, and the rest will be a mass of nothingness. So that's the IDR, intrinsically disordered regions. But they shift shape in the context of interaction. So they change their shape. That makes them very, very critical for interaction, and very important also for the evolution of new interactions. Maybe it's a way to evolve new interactions and new functions. And more than 30% of human proteome is predicted to be intrinsically disordered. That also is an indication that they are everywhere and they are important. And eukaryotic transcription factors are especially enriched by these regions. And here is the Drosophila bicoid. As you can see, this is the homeodomain, highly structured, and this ST-rich, glutamine-rich, alanine-rich, that's how they're called, with the enrichment of the residues in those regions. So because they are not, nothing is known about what they, well, of course, ST-rich are important for phosphorylation. Things are known, of course, but about the structure, not much. So they contribute to in vivo transcription activity, or that's what predicted to be how they predicted to contribute via DNA search and binding. Basically, they make very weak contacts with DNA and protein, so that might be enhancing the transcription factor binding its target sequence. They might be involved in protein-protein interactions, and they might also provide surface or motifs for cross-translational modifications. They might also lead to chromatin conformation changes, and they also can be involved through all of these. They might change the transcriptional kinetics, which is very important because temporal regulation cannot be really detected by the static and biochemical methods, in vitro methods. And I'll try to address these questions in bicoid or in Drosophila by using again bicoid. And this time IDR mutants, we are making these different kinds of mutants and trying to understand if they affect bicoid developmental function, DNA binding, chromatin conformation, protein-protein interactions, and transcriptional dynamics. And based on this data, and also doing some computational analysis in order to understand is there a pattern to these IDRs? Can we design a functional IDR and trying to construct a synthetic bicoid protein with the homeodomain and some IDRs? So those are the things that we are going to do. And finally, this is the last slide, I promise. And this is something that I'm very excited about. That's a very I think very straightforward. I mean, very difficult to do, needs a lot of work to do the experiment, but I think it's a very telling experiment. So we are going to, so these are, as I said, these are isogenic lines which have 10% of survival rate. So we want to understand what sets these survivors apart from the rest. And it's not genetics. I think we haven't shown that, but we repeated this experiment, and to get some random changes every time is kind of very highly unlikely. So we want to find what these non-genetic factors are, and if they are inherited or not. And maybe if this is the bottleneck of evolution, this might be what Weddington somehow tried to do, or that he did, like that would lead to evolution of novelties through this maybe phenotypic changes first that doesn't involve genetic changes. With that, I would like to thank all the people who helped me with this work that I presented here today. All the people, Steve Small, my postdoc PI, and Shelby Blight in Northwestern, who I also worked with and with whom I did the ChIP-seq analysis, and the Thornton and Schmidt-Ott labs in University of Chicago, NYU, Steve Small. And in my lab, Dilan is my first master student, first ever master student, and my undergrads. And Bilkent and BG, especially Pelin Makas, our lab manager, who really is wonderful. She did a lot of work for our lab, for me, for my flies. So I'm thankful to her. And also I would like to thank Ergiden Zosoy's lab, especially Mehmet Gözberg, and I can't really overestimate his help and all of these people's help for us opening up their lab for us, and from the very first day. So, and that's very important. Because there are not so many Drosophila labs in Turkey, and it was a big it was a little bit naive for me to think that I would come here and start right away, and I hadn't come here thinking what will happen, but I really had the amateur chance, if you might I got very lucky being just coming next to Hacettepe University to the great lab. And thank you all for watching us live or not. And if you have any question, I'll be happy to answer.
Teşekkür ederiz Pınar. Oldukça ilginç, güzel bir sunum.
Ay,
yoğun bir
çok...
konu.
Biraz çok mu uzatmışım?
Yok, gayet iyi. Şimdi ben bu arada sorular...
5 dakika diye düşünüyordum.
Yok, gayet iyi. Sorular gelinceye kadar insanlar chatten yazacaklardır. Ben de o arada şey yaparım sana. Onlar gelinceye kadar soruları okurum geldikleri zaman. Orada birkaç şey ben en azından sormuş olayım. Eminim izleyenlerimiz, dinleyenlerimiz arasında tabii bu konulara oldukça uzak olan ama bir taraftan çalışmak isteyen kişiler de vardır. Dediğin gibi böyle bir Drosophila araştırması, özellikle developmental olarak EVO Drosophila araştırması yapan laboratuvar kaç tane ki zaten Türkiye'de herhalde yeganelerden bir tanesi. Böyle bir araştırmacı da olacaksın burada. Hem belki diğer ilgilenen kişilere de bilgi verecektir diye. Birkaç belki basit soruyla ben başlayayım izninle. Örneğin bu konstruktlardan bahsediyorsun ya, mesela slaytların hazır burada dururken şeye doğru dönebilir miyiz? Öyle en baştaki slaytlara şöyle çünkü örneğin evet geriye doğru belki yukarılarda şöyle ne diyelim biraz daha aşağı inecek olursak. Örneğin mesela o güzel bir slayttı galiba 10. slayt falan olabilir. Burada için ilk koyduğumuzda örneğin bir zen, diğer tarafta bir koyut var, homodomain region. Bu bir construct diyorsun. Bunları ilk başta nasıl oluşturuyorsunuz ve bunları egg safhasında mıyken Drosophila'ya veriyorsunuz? Biraz bunları açabilir misin?
Haklısın. Şöyle bunlar plazmid dediğim gibi bakteride ekspres ediyoruz. Önce bunları klonluyoruz bakteride. Ondan sonra büyüttükten sonra embriyoya transfect ediyoruz. Drosophila embriyosuna. Drosophila embriyosunun şöyle bir özelliği var. Syncytium. Yani hücre membranı yok henüz. Daha bu nuclear cycle ya da çekirdek devrinin 14. safhasına kadar henüz sel membranı yok. O yüzden erken bir safhada yakalarsam ve posterior kısmına enjekte edersem bu construct'ı ve aynı zamanda bu construct'ların bir de recombination site'ı var tabii ki en önemlisi. Bunlar naive bir embriyoya da inject edilmiyorlar. Bunlar daha önce transfect edilmiş ve bu transfection'ı da key element daha önceden yapılan virüsün recombination site'larıyla yapılan bir şeyle entegre edilmiş rekombinase olan embriyolara enjekte ediliyor ve benim o kısa recombination site'larım genom üzerinde belli yerleri tanıyor. Gene bunlar Drosophilanın bir güzelliği de yani hani bu landing site dediğimiz şeyler, yani o recombination site'larının olduğu ve sen basitçe mesela bu construct'ı ben düzenliyorum. Bu construct'ta ben bu 40 base pair uzunluğunda 2 tane koyuyorum iki yanına. Bu maternal'ın mesela 2 tarafına bu recombination site'ları koyuyorum plasmitte. Bu recombination site'ları genom üzerindeki recombination site'larına ne denir ona? Complementary. Ve o da bir transgenik bir zaten embriyo. Benim bu transfect ettiğim. O transgenik embriyonda recombinase var. Ben inject ediyorum embriyoya. Embriyoda hücre membranı yok ve posterior kısmında da germ seller oluşacak. Posterior'a inject ediyorum. Oraya posterior'a doğru giderken çekirdekler daha hücre zarını, onlar hücre çeperini daha önce oluşturuyorlar. Oluştururken benim DNA'yı da içleyen alıyorlar. Ve orada recombinase de gene germ cell'de express edilen bir recombinase. Germ cell'e entegre olan şeyler bir sonraki o embriyodan büyüyen adultlar bu sefer benim başka bir sineğe çaprazlıyorum. Ondan sonra onun offspring'inden bazı markerlar koyuyorum tabii. Göz markeri mesela bu construct'ıma. Öyle seçi veriyorum. Yani bu çok yani hani bir sürü bunu construct'ı yapmaktan embriyoyu almaya kadar geçen süre transfected ya da transformant almaya geçen süre 2-3 ayı buluyor tabii. O kadar basit bir şey değil. Yani her şey yolunda giderse tabii. Ama her şey possible. zaman ve güzel. Emek istiyor tabii ki.
Tabii. Evet. Güzel. Bu bayağı açıklayıcı oldu. Sağ ol. Hani nasıl bir anda bunların yani elde edilmediği aradaki stepleri de aynı zamanda görmüş, anlatmış oldu. İyi oldu. Evet. Sağ ol. Yine tabii bu arada hani teşekkür ve oldukça güzel sunum olduğuna dair hani yorumlar var. YouTube üzerinden onları da söyleyeyim. Bu arada biraz daha aşağı inersek hani şu ancestor olması konusunda bir şey göstermiştin. Filogenetik ağaç, belki onun üzerinden evet burada hani bir konuşabiliriz. Öncelikle şeyi sormak istedim. Burada görselde bir tanesi Dimel, Drosophila, Melanogaster var, MDO. Bu yani ancestral bikoid ve ancestral bu ZB noktası gelinceye kadar. Şimdi hepsi bir farklı Drosophila türlerinden bahsediyoruz, farklı canlılardan bahsediyoruz. Biraz burada hangi organizmalara baktığımızı açıklayabilir misin bu ağaçta?
Tabii ki. Şöyle, bütün drozophilaların bikoid homeodomain sequence, bunlar homeodomainleri bu arada. Sadece homeodomainlerine göre phylogenetik ağaç kurulmuş durumda. Bütün drozophilaların ya da elimizdeki sekansları olan drozophilaların bütün homeodomain sekansları aynı olduğu için bir tane homeodomain sequence aldık. O da Drosophila melanogaster diye geçiyor. Mesela Megacelia'nın var MAB, Lucilia, Califora, LSI, Lucilia sericata, Califora Stinia, şuradakiler mesela. Bikoid sadece işte bu burada gösterdiğim 8-9 canlıda bulunan bir şey olduğu için sadece oradan bikoid sekansları alabildik. Bunlar dediğim gibi ev sineği, at sineği gibi öyle sinekler, 2 kanatlı sinekler. Onun dışında zen, ama zen daha geniş bir şeyde bulunuyor, ama biz zaten bunlarda bulunan zenleri aldık. Bir de onun üzerine başka şeylerde, başka inseklerde, başka sineklerde bulunan ve bikoidi olmayan sineklerde bulunan zenleri de aldık. Ondan sonra hatta tribolium'a kadar gittik. Ama mesela Hoxtree'de burada out grup olarak kullandık. Çünkü Hoxtree'den derive olduğunu biliyoruz zenin ve bikoidin. Onları da out grup olarak kullandık bu ağacı yaparken. Bunun detayları tabii benim çok da hakim olmadığım şeyler, ama bunlar en basitten benim anladığım kadarıyla bu homeodomain sekansları alındı, bu insect ve değişik fly türlerinden. Ondan sonra bir de bunların zaten insektlerde ve sineklerde phylogenetic relationship ya da ilişki zaten bilindiği için sadece homeodomain'e bakılıp bir ağaç yapılması çok da güvenilir olmayabileceği için bu homeodomain homolojisi ya da işte ona bağlı ağaç, bir de üzerine filogenetik, daha önce bilinen insek filogenesinin üzerine oturtulup maximum likelihood'la en uygun ağaç, en uygun şey nasıl diye seçildi. Dediğim gibi bunları Evolution Organismal Biology Departmanı'nda University of Chicago'da Joe Thornton'ın bir postdoc yaptı ve onlar bu işi, yani artık gündelik onların işi böyle daha gene familleri falan çalışıyorlar, işte receptor family, steroid receptor family falan filan gibi daha fazla family'de olunca böyle daha fazla nodla falan da çalışıyorlar. Burada sadece 2 nod olabildi çünkü o kadar çok fazla tarihi o kadar çok fazla dallanıp budaklanmadığı için bildiğimiz bu kadar tür ya da bu kadar homolog olduğu için de bu sadece bu 2 branşa kaldı.
Evet, güzel. Ne kadar en azından burada da ne kadar korunmuş olduğunu görüyoruz. Oldukça ilginç. Ancestral Zen'den sonra ne kadar hızlı bir evrim geçirerek bikoide uğraştığını göstermesi de çok ilginç. Bu da der misin, herhalde bu residülerin, homeodomain'de olan bazı residülerin ne kadar önemli olduğundan bahsettin. En azından 11 tanesi benim de anlayabildiğim kadarıyla önemliydi. Keza herhalde burada bir nevi sadece teker teker böyle tabii rastgele mutasyonlardansa birbirini komplemente edecek, yani düzgün değil mi? Fonksiyonu koruyarak ilerleyebilecek herhalde değişimler oluyor ki senin de gösterdiğin gibi, yani en ufak bir amino asitin değişimi, örneğin 50. pozisyonda olduğu gibi bu işi tamamen lethal olabilecek bir şeye çevirebiliyor. Burada hızlı bir evrim var çünkü ancak buraya sayesinde zaten bikoid kendi düzgün fonksiyonu yapabilir halde oluyor herhalde, değil mi?
Yani çünküsünü bilemiyorum tabii. Neden bikoid evrildi? Ama ya da zaten bu yolla olduğuna dair de bizim bunu kanıtlamamız için fosil kanıtı vesaire gerekir. Tabii böyle olduğunu bilmiyoruz. Ama bu olurken aynı zamanda zaten bu değişimler kafayı hala yapan bir sistemin içinde gelişiyor. Bir gene duplication olmuş ve bunlar gelişiyor. Bu acaba gelişirken mesela K50 olduğu bir kısmını mı aldı acaba? Diğeri kaybederken bu protein mi aldı? Çünkü bu ölü olsaydı zaten orada bir deden olurdu. Bu süreçten geçebildi çünkü bir cin duplikasyonu vardı. O hala kafayı yapmaya da ya da yani hani düzen yapmıyordu da o sırada başka bir kafa proteini zaten vardı. Yapmaya devam ediyordu. O ancestral'dan buna geçiş nasıl oldu o kısmı bilmiyoruz. Değişik tezler var tabii ama hala bir kanıt yok. Herkesin bir tezi var bu konuda. Ama.
Evet, evet, ilginç. Ben de bir bakayım başka sorular var mı diye sana sorabileceğimiz gelen. Evet, başka şimdilik soru da yok. Herkes ancak sindiriyor herhalde
Çok
buradaki
mu ağır
bilgileri.
oldu?
Evet.
Biraz
Evet, yani
uzattım
bence oldukça.
sanki, biraz çok
Yo, yo,
anlattım.
yok, aslında hani açıklamak için
Bulmuşken sahneyi
tabii.
anlatayım dedim.
İyi yaptın, evet. Yani zaten anlatmak gerekiyor. Hani bir anda herkesin aşina olduğu konular değil, bahsederek bol örneklerle vermekte iyi oldu. Peki o zaman tekrar bakayım. Başka peki sorularımız da yok. Teşekkür ederiz tekrardan
Ben
katıldığın için.
teşekkür
Bizde de
ediyorum. Çok
kendini
sağ olun. Çok
laboratuvarını
teşekkürler.
tanıttığın için. O zaman buradan şey yapalım, yayınımızı sonlandırabiliriz. Herkese katılımları, dinlemeleri için de teşekkür ederiz. Sorularınız olursa Pınar Hoca'yı nerede artık bulacağını biliyorsunuz. Kendisi de Bilkent'te, onunla iletişime geçebilirsiniz. Eminim öğrencileri var, yeni öğrencilere de ihtiyaç duyacak. Belki siz de bu konularda çalışırsınız. Türkiye'de bu konularda çalışan yegane araştırmacılardan olursunuz. Peki, ben şimdi